Hello, everyone. Good morning. So my name is Jian from Carnegie Mellon, and first talk today. The title of my presentation is Large Language Models in Computational Biology. So I would like to give you a very brief and also high-level introduction on first of all, what is large language models? What is language model? And what constitutes a large language model? Like some of the concepts. And certainly I will go through some of the very recent development in this field in terms of applications to genomics. And as you know, everyone is talking about LLMs and large language models. Like most people, I don't know. I'm actually not sure I know exactly what I'm talking about. This is certainly a very fast-moving field. There's a lot of new developments and ideas. So this is not a research talk, per se. This is also not a tutorial. I hope this is more like a journal club that can spark some discussions with all of you in the audience, certainly beyond the duration of the talk. Anybody know who this is? This is a Dutch man, Antonio van Leeuwenhoek, and lives in the late 1600s, so about 350 years ago. He was actually a cloth merchant, selling clothes and fabrics. And he likes to assess his, cloths and fabrics. So he built these magnifying lenses. And so this is the tool that he built, in late 1600. And one day, he pointed his magnifying lens to a droplet of water. And he saw something as moving. So now we all credit to Leeuwenhoek that he was the first who first observed a living cell that was 350 years ago. So this, as I mentioned, marked the beginning of observing living cells. And of course, until today, we don't have a lot of understanding of how the cell, first of all, is being organized. We have some idea, but a lot of room for new understanding, and certainly we don't know too much about how the cell works. So this story tells us a few things, right? First of all, Leeuwenhoek is very interdisciplinary. He was a cloth merchant, but later on, he developed this interest to observing living cells. Second, the role of the tool, right? You need a tool to look at things. Third, anyone? Don't be afraid of pointing your tool to something unexpected, or if you see something unexpected, follow paths there. Feel free to follow paths that even they're less taken. So I think it's a interesting story. So keep in mind, I will get back to this in terms of our understanding of the cells and how to use these new technologies. Right? All of you are working on computational biology, and computational tools and techniques and algorithms is your tool in your hand. Even though I don't have time to talk about my own research, so this slide basically summarizes what we do in our own group. So we have tremendous interest to develop computational methods to model the multi-scale structure of the cell. And these days, everything is in single cell. And look at these, how the genome is being organized in three-dimensional space within the cell nucleus. You have heard Christina Leslie's talk yesterday, and it was relevant to that. But in the nucleus, in addition to DNA, there are also other biomolecules, how they're being interacting with each other. And if you step back, you will see, cell nucleus is just one part of the cell. There are other organelles and components in the cell. And on here, this is a recent work from the Allen Institute for Cell Sciences, where they use the live cell imaging to build this model where you can see different structures within the cell. And if you keep stepping back, you will see these cells are also organized in complex tissues, and how we have heard quite a few talks already on spatial genomics, spatial transcriptome. This is a very exciting area, and my group, we have also been developing methods like this recent paper in Nature Genetics where we try to model the cell as a collection or a combination of underlying factors. Just like so the tool's name is called spice mix. The rationale of naming that spice mix is just like spice mix. If you put different proportions of the spice together, you get different flavors. And the cell might be working in similar ways where you put different strengths or weights for different underlying factors, the cell may behave in different, they have different identities. But I think ultimately, our long-term goal is to have a very comprehensive understanding of this multi-scale organization, especially in single-cell resolution, right? And then, so I saw this year, so this Oscar-winning movie nicely summarizes our goal. So everything, everywhere, all at once. If you can build a model that can captures every single component within the cell all at once to have a very comprehensive view, that will be fantastic. So last year I showed this slide. This is the CGSI YouTube. Ben mentioned this in his talk, and it reminded me. So I actually showed this slide last year. So this is all the CGSI talks on YouTube channel. If you rank them by popularity, and last year I found out that I was ranked number 3. Shuyan was number 1, Dana was number 2, I was number 3. And the other day, Ben, just during Ben's talk, I was like, I need to check it out again, right? So see how things have changed. And take a guess. What's the ranking today?
You were first?
No. But I was second. So so if you look at the numbers, the CGSI channel has more subscribers. And for some reason, in the past year, there were almost like over 2,000 people have watched, right? Over 2,000 people have watched my talk. This was actually 4 years ago, a talk. And Sriram's talk, is way ahead. It's like almost 15,000 talks. So this is kind of an interesting finding. So I think the message to all the students in the audience is that there's some talks from previous years and worth checking out. So as I was asked to present this talk, and then I was like, I can do that, right? It's how hard that would be. And then, we talk about these things all the time. And the more papers I read, I during the preparation, I realized this is actually quite a difficult task to cover a lot of materials. And I also talked to my students. I just said, hey guys, I'm giving this talk in UCLA on large language model and computational biology, and you guys need to help me. And I also realized that actually all my students know more than I do. So that's what I learned from preparing this talk. So it's a very humbling experience. So I asked them for help. So this talk was actually put together by 3 people in my group, Ellie Haber, who is here in the audience, and Wendell is also another graduate student, and Xiaohun is a postdoc, so he just graduated from Loai Necklace Lab from Rice, and he's a postdoc in my group. So this talk is really a lot of effort from these 2 people in my group. So let's get to the real material, the actual materials for the presentation. So first, I would like to give you a recent history of language models. And this is not a new thing. This has been there for decades. Previously, there's tons of work. It's called statistical language models, right? People use probabilistic models, try to model natural languages and try to predict the next word based upon most recent context, using some Markov assumptions, for instance. And I listed some references at the bottom. For instance, this first one is actually written by my CMU colleague, Ronnie Rosenfeld. He previously was a very active researcher in this field. He wrote a very nice review over 20 years ago. So if you have interest, take a look at the review, and then also the papers there, because I'm sure the papers are all older than most students here. But reading papers that are older than you is not a bad idea. And second, later on, there's these neural language models, right? You can try to derive the probability of word sequences from neural nets. And famous examples like word2vec, right? And then, in Christina Leslie's talk, she actually touched upon some of those concepts, like their previous work, bind spaces. There's some connections to these neural language models. And more recently, these, really started at the beginning of 2000, there are these notion of, so 2000 is the neural language models, but these pre-trained language models are more recent effort. So you can try to capture the context of word representation through pre-training. So if you pre-train a model and then fine-tune a model for downstream tasks. So famous examples like Elmo, the pre-trained with bidirectional LSTM, and more recent models like BERT and GPT, which I will elaborate a little bit later on. And they all based on a model structure called transformer, which I'll also elaborate a little bit more. So these models, essentially, you can pre-train on large amount of training data to derive the representation of your, let's say, words, tokens, or sentences. And then you can use them to fine-tune your model for specific downstream tasks. That's the general rationale. So the last one is a tutorial that I used, a tutorial article that I used to put some of these slides together. So if you have interest, you can take a look. So what is large language models? Large language models, in essence, is just very large-sized pre-trained language models. So we have seen pre-trained language model. You need to pre-train a model with large amount of data, and then you fine-tune that for specific purposes. And large language model is a very large-sized pre-trained language model. It's still confusing. What does that mean? Large-sized. Like, what constitutes large? It is actually evolving term or concept. Some people say these days at least you need to have 50 million parameters in the model, and a lot of money to train them. And the model has some unique properties. For instance, the model can really find out some, like what they call, at least at the bottom, these surprising emergent abilities. So in these large language models, there's a quite well-known observation of phenomena called scaling laws. And the paper, this Kaplan et al. From OpenAI, they published this empirical analysis, basically, is to try, if you have limited allocation, right? You have or as you in terms of compute budget, how do you allocate that? And they find out that to optimally allocate the resources, you need to dedicate the resources to building a larger model rather than collecting, applying this on more data. And that's really the motivation. A lot of these recent development, building large models so that the model can have more powerful capabilities. But in the meantime, there's certainly other type of opinions. For example, this Hoffman et al. This is in last year in Europe, and I think this is from DeepMind, and they have some different analysis and find out that the data itself is also equally important. So you really need to balance the data and also the model size at the same time. So if you compare regular language models with large language models have some very unique characteristics that people expect to see. First of all, as I said, is the number of model parameters. Tens of millions, tens of billions, and hundreds of billions of parameters. And I think that the recent Meta released the Llama 2 the other day. It was like over 70 billion parameters. And the large language model need to have some kind of so-called emergent abilities. Is that when the model reaches a certain level, it can do things that smaller model cannot do. So there is a turning point, some kind of a turning point somewhere where the model starts to behave in a very powerful way. And also the large language models, as we all know, the GPT-4, they have these APIs. You can through these prompting, you can actually derive your own applications and models. This could be also a very important aspect of that, and large language models need tremendous resources to build. I just found out that this is a recent announcement from this company called Infection AI, and they said, oh, it's big week. We have raised the 1.3 billion and are building the world's largest AI cluster, and 22,000 H100. So the current price for H100 is about $40,000 to $50,000 retail price, $40,000 to $50,000. So if you do the math, this is over $1 billion, right? Just to build the GPU cluster. But you know, NVIDIA is an investor. I'm sure they get some discount, but still, this is a large amount of resource just for building the compute cluster. And they're certainly following the scaling law closely, right? So most of the resources is dedicated to building large models to have these large number of H100 to enable building a very large model. And then there's also this term called foundation models. What is foundation models? The large language models could be could be a foundation model, right? So foundation model is basically when you train a model, you can repurpose or fine-tune this model for different kind of tasks. Like you can train a model through text, media, image, visuals, etc., and you build this foundation model where you can use the model to do different things. So in this article from the Stanford Center for Foundation Model Research, I think it's a good review, and they basically talk about all different aspects of foundation models. And again, this follows this rationale of pre-training on large unlabeled data set in self-supervised or unsupervised way, and self-supervised way, and then you can fine-tune the model to transfer that to different contexts. And well-known examples like GPT and BERT, etc. So let's talk a little bit about this transformer, because this is really the basis for all these large language models, almost all these large language models we have seen so far. The reason to develop this transformer, by the way, this is the paper that introduced this transformer architecture in Europe's 2017, and in the past 5 years, it has been cited over 80,000 times. So the goal, the rationale of developing transformer is that we need dynamic representation of context-specific information, like to statement like it and I do not like it. The word like has different meaning, and it should have different representations from these contexts because it's in different contexts. Also, oftentimes we want to know in a sentence like how these words are, what's their connections or dependencies among them. And I'll get to that because in biology, sometimes we also try to understand those dependencies, right? A genetic variant right here, what's the connections to something elsewhere in both in close proximity or even in somewhere far away. And previous models like RNN or LSTM or CNN have their own issues, and transformer combines the benefit of dynamic computing, good memory, and parallelizability. So just a little bit more on the structure. What is a transformer? So this is really designed to handle sequential data like text or time series data, and it has the advantage of capturing long-range dependencies and context. Like you have a long sentence, or you can model that, and previous models tried to achieve that, but they all have their own limitations in terms of the context they can cover. And it utilizes a key mechanism called self-attention. Essentially is to look at each token or each word in order to, during the training, like what are the other words that this word attends to, right? So you can consider that as the dependencies among these words and their connections and the underlying meaning of the sentence that we are looking at. And the model consists mostly two parts, the encoder and the decoder, and both contain some core blocks of attention and a feed-forward network here showing on the slide. Just a bit more here, let me see. Yes, so in this attention, what is attention, right? In the transformer, there's this concept called attention. What does that mean? And how it is being calculated? So the brief version of attention is basically a scaled dot product attention that is being used in Transformer. And this can be described as a mapping, a query and a set of key value pairs as an to an output, where the query Q here, keys K, and the value V, and also the output they're all vectors. So showing here, this is just one scaled dot product attention, but in transformers, these attentions can be they use the so-called multi-head attention so that you'll be able to comprehensively learn the dependency between the token in the entire input. And so multi-head attention basically consists of several attention layers running in parallel, and it allows the model to jointly attend information from different representations at different positions. So the essence of the attention is summarized here, right? So there is a matrix multiplication of Q and K, and there's a scaling factor. This is the dimension of K, and V is the value, and there's a softmax that applies to this after the Q and K matrix multiplication. So so here's, as my student pointed to me, that there's actually a quite good visual illustration of how the attention works through the link at the bottom. And if you have interest, you can take a look. For example, in this sentence, the animal didn't cross the street because it was too tired. What it refers to, right? It refers to animal, and these kind of dependencies can be learned from natural language. And when we look at these natural language sequences, you can think about like what's the connections to our genomic data, right? Or each token is a k-mer sequence, or each token could be a gene, and what these kind of attention actually mean, right? So if there's attention between two different genes, what does that mean? Or attention for a particular gene, a gene has higher attention, what does that mean? So we'll get to that because there's some recent development exactly utilizing these properties that can be learned from the model. So the transformer architecture in BERT and GPT, because these are the two that most widely used in genomic applications. So for the BERT, most of them use encoder only or encoder and decoder. And for the GPT style models, they use the decoder part only. And the goals are a bit different. For BERT, you want to train a masked language model. I have another cartoon on the next slide showing what that means. And the model type is discriminative, and then pre-trained task is to predict the masked words. So basically during pre-training, you mask the words and try to use the rest of words to predict the probability of a word showing up at a certain position. For GPT style, it's a bit different. It's called autoregressive. It means that it tries to predict the next word using the words prior to that. Right? So if you have a sequence and you see the words that are prior to that, and you try to predict the next word based on what you have previous. And then the model is generative, and then so the task is to predict the next one. Now here's the cartoon that I mentioned for the pre-training class. For the BERT style masked language model, you mask the word and then try to predict the probability of this word. Like this is your input, and you mask it during your pre-training, and you mask this word, and then you train a model to train the probability of a certain word showing up. You say, oh, the probability of you here is the highest, and so this is the part of mask, and then pre-train. For autoregressive is you train a model where you can predict the next word using the words that you have seen. So essentially is in a sequential manner for the words that you have seen. So the chicken crossed the road. Road is what you try to predict. And this slide from this figure from this recent review article, it summarizes all these models, like where they use the decoder part or use the encoder and decoder together, or only the encoder part. And you can see that there is a trend where these models are, many of them are using the decoder only, including GPT here, and then the Meta Llama, and then the Google BART. And on the right-hand side listed, these are the number of parameters that are being used in these models, like GPT-4 has 1.7 trillion parameters. All right, so let's get to the second part, is the applications of these models in genomics. And these large pre-trained models can be utilized for fine-tuning for or on downstream tasks with limited training data, right? So that's certainly one motivation that these kind of models can be used in biology, because we know that in many contexts, data itself is sparse and noisy in certain conditions. You actually don't have that many data sets. The data sets is also incomplete, like rare disease or very precious samples that you cannot get. So the notion is that if you can, through these pre-trained models, you can train the embeddings of your entities, either it's genes or sequences, that can be generalized to many different contexts through some additional fine-tuning. That would be fantastic, right? And that's really the motivation of two directions that I want to talk about. There's also a lot of development in protein structure and function modeling, which I won't cover today. I will talk about topics that are more relevant to genomics. So one is on modeling the genome sequences, and the second part is modeling the single cell, gene expressed single cell data that we have heard a lot about this week. So first, these large language models for genomic sequences, I listed a few of these models: Big BERT, DNA BERT, which I will elaborate a little bit. And there's also in this informer, it's debatable whether this can be considered a large language model, because it doesn't, and some people think it doesn't really count as a language model, because even if it uses the transformer architecture, it doesn't really model the probability of like a sentence, like what language models does, like the sequential dependencies of the tokens that you're looking at. And there are some other, some more recent development. So what is DNA BERT? DNA BERT, it exactly uses this BERT architecture, pre-trained BERT for DNA sequence based on the human reference genome that they used. And so in all the models, all the models that I go through today, in my view, in my personal view, I think the most important task in these modeling endeavor is the tokenization. Tokenization means that how you represent a word in our genomics context. There are other very important steps, but I feel like that's a very important step. And right now, their DNA sequence or single cell, they have their own way of tokenizing these entities. Whether or not that's appropriate, I think is subject to further research. For the DNA BERT, they just use k-mers as tokens. And these k-mers are overlapping. So which means you have AAT, and then the next word is, next token will be AT something. So right, it's overlapping, right? K-mer, and then the overlapping part is k-1. So here's on the right-hand side, this is a DNA BERT structure. Is almost identical if you look at the bottom part. So essentially, you can tokenize this, the original sequence, right? This is the sequence. You tokenize that. So there's some other tokens like the classes of the sequences or separations, etc., like AGC, GCA, like overlapping k-mers. So during pre-training, this is the BERT, right? You need to mask something, and you try to recover that. Make sense? And you can mask that. And so the author said, oh, you need to consecutively mask 3 k-mers at the same time. And my question for you is, why do we need to do that? Why do you need to mask 3 k-mers consecutively rather than just randomly mask? Because you're overlapping. Because if you just mask one, you can kind of guess what it is trivially, right? Because you have neighboring. So that's, you need to. So in summary, if you don't do that, and there's information leakage already in your given data, so that the training, et cetera, becomes trivial. There's no point of doing that. So that's the in tokenization in DNA BERT. And the authors of DNA BERT had a very recent update. I think this is just like few weeks ago. They call it DNA-BERT 2. So they try to use another way to tokenize this. So in the paper, they actually talk about this different kind of tokenizations. So the one that they used in DNA-BERT is overlapping k-mers. And they say, oh, there's some disadvantages, even though you consecutively mask k-mers, you still may have information leakage, at the beginning and end of the sequence. And if you don't do overlapping k-mer tokenization, if you do non-overlapping, and that's even trickier because if it's non-overlapping, like example here, this example, you have two sequences, and if you just remove one character here, A, and these two sequences actually very different, will have very different tokenization, and then all these tokens are completely different. So it's very inefficient in a way. So the authors use this approach called, in DNA-BERT 2, it's called byte pair encoding. It's kind of some kind of compression trick. So in first iteration, you only have 4 characters in your alphabet. In the next iteration, you try to count what is the most frequent, like in this case, two letters, right? So in this case, and that becomes part of the characters. So TA joins the vocabulary, and then you continue. And so eventually you're going to have a list of tokens or n-mers with varying lengths, and those will be used for their tokenization. And they have some other tricks to make the computation more efficient. But I think if you look at their results, the results are a bit confusing, I would say, because in different tasks, the advantage doesn't seem to be that dramatic. Sometimes it's doing better than DNA-BERT, and there's another recent model called nucleotide transformer, and also it uses transformer architecture. But you can see that these different kind of performance listed below, they have varying performance. I think this is still a very, how to say, very much unsolved problem, how you represent the DNA sequence in terms of tokenization in order to apply these transformer-based models. All right? So this just very briefly, genomic level application of these language models.
So like, I'm just looking at those results. The COVID variant classification, that seems to be a really easy problem, but the performance is like 55. Is that like 55% or something like that? I'm just curious.
Oh, I don't know. I can't answer that question.
Bad, right?
Yeah, it's not too impressive. I know.
Yeah.
All right. So now this is genomic sequence, and then a few more examples on single-cell genomics. And these are I selected I think there are a couple more, but these are the ones that people have developed for modeling single-cell data. And SC-BERT, this was published last year in Nature Machine Intelligence. Machine Intelligence. Yes, Nature Machine Intelligence. And there is also a recent paper on Nature, a method called Geneformer. Okay? SC-BERT, as the name says, it uses the BERT architecture. Geneformer, I believe, also uses BERT-like structure. And SC Foundation, there's a more recent model called SC Foundation. If I have time, I can go through that. But the SC-GPT, as the name says, uses the GPT model. I think this is from Bo Wang's group from Toronto. I heard there's students from both labs, so feel free to chime in if I'm not stating things properly. So let's first look at this SC-BERT model, because this is the first, I believe this is the first model published, sort of using these transformer-based large language models for single-cell modeling. And it learns, first, pre-trained, right, on lots of single-cell RNA-seq data. And again, remember what I said, tokenization is important. How do you represent single-cell RNA data as words and sequences? How? How do you do that? So in almost all of these methods, they view a cell as a sentence and a gene as a word. All right? So the next question is, how do you represent a word in terms of expression, because expression is continuous and a lot of noise and so forth. And how do you represent that? So one way to mitigate that is to do binning. Expression, you chop it up into several buckets and say, oh, this gene expression is very high, kind of high, medium, low, and so forth, right? In general, not exactly what they did, but that's really the concept. So sort of to discretize expression values to produce these. Also, you can turn that into an embedding. So the authors did, that's for the expression embedding. That's the first part, the first row. And then the second row here, they also try to somehow capture some intrinsic properties of gene-gene dependencies. Like we all know, these genes in our cell, they're not individual entities. They kind of depend on each other, right? So a gene goes up, the other gene may go up as well, because there might be some underlying regulatory relationships between them. How do you capture that? There was actually some, many methods like that. You try to represent a gene, like a generative embedding, according to co-expression or their positions in a complex gene regulatory network. So the authors used a previous method called gene-to-vec, right? Basically turn a gene into a vector, into embedding, by incorporating their gene-gene interactions in many different biological contexts. So essentially, they aggregate these two types of embedding for each gene. One is the gene-to-vec embedding that captures to some extent the gene-gene dependencies, and also the other is the capturing the embeddings of the expression from each gene in that particular cell. So that part is the pre-training. And then again, you remember that we talk about the BERT, right? You mask something and try to recover that, right? And you can mask a gene expression and try to use the rest of the gene expression to recover that. And then the model is trained that way. You can use the self-supervised pre-training to achieve that. And once you have the model, once you have the pre-trained model, you can fine-tune that for different tasks. For example, cell type classification. If I give you a gene expression, if you give you a single cell with the gene expression, can you tell me which cell type this cell belongs to, right? So this is a common task in single cell analysis, and you can also fine-tune this model to achieve that. I'm going to skip these slides because I think I already explained this. And if you read the paper, and of course, I skip all the real data results because I think for the most part, for all these methods, I would say it's fair to state that all these methods, I think the models are very interesting, but the improvement over existing models is moderate. Sometimes it's comparable, sometimes even a little worse, sometimes a bit better, but overall, I think it's moderate. And we could get to that in terms of, what's the future. And in another recent method, Geneformer, they also train on many different single-cell RNA data set. They discretize expression so the tokenization here is that the token is still the genes. But because of, you try to model the context, right? And what they did is they ranked the genes according to their relative expressions. So imagine you have many single cells, many cell types. In a particular single cell, you look at a certain gene expression compared to its expression in the rest of the single cells. And if the expression in that particular single cell is much higher than the rest of the samples, then the relative ranking is high in that particular cell. Does that make sense? So essentially, you have a ranking. So the value is not necessarily expression, here according to, but it's the ranking of the gene in that particular cell. So housekeeping genes is expressed all over the place very highly. So it's not going to be ranked very high in a particular cell because it's simply expressed very highly all over the place. For some, even for some, transcription factors, even they're expressed sometimes quite low, right, in certain cells, but they could still be ranked high. Why? Because even they're relatively low in that particular cell, but compared to the rest of the cells, the expression is already high. And it's a way to calibrate and reorganize the rankings of the cells and give them the quantity that you can operate on later. And so and then the authors, like one thing that I think they did is very interesting to look at the attention weights from the models and found that these attention reflect important genes such as the gene nodes that are in the regulatory network that is the central regulatory genes versus the more peripheral ones, and the more central ones will have higher attention because they kind of influence a lot of other genes in the cell. And you can also train a model to do in silico perturbation, meaning that you can remove a gene in silico computationally, and you assess its impact. And how to assess its impact? You remove a gene, and you compare the embeddings of the cell and also each individual gene before and after you remove that particular gene. If the embeddings before and after are drastically different, it means that that particular gene may have some very detrimental effect in that context. So that's Geneformer. It's interesting. And this is the last example I want to show. This is the SC-GPT, again, from Bo Wang's group. So they use this, again, they trained this pre-trained on over 30 million normal human cells from many different tissues, and try to learn insights for genes and cells, and follow the same steps that we have seen over and over again in these models. You pre-train on large number of samples, and then you fine-tune that for your specific tasks. So they did this for a number of tasks, cell type annotation, multi-omic integration, perturbation predictions. Again, tokenization, right? How they represent each gene. So they also discretize the gene expression values, like the blue bars here. And you also add gene tokens and also condition tokens. The conditions means different batches or whether it has perturbation involved in that particular cell. So maybe very, very briefly, I think one interesting aspect in SC-GPT is the generative pre-training. So this is because it's GPT, right? It's auto-progressive. It's you try to use the information that you have seen to predict the next word, like in natural language. And here in SC-GPT, the authors came up with this idea that you can try to predict the what they call unknown genes from the known genes that you have had. So in a way that, and then you do this iteratively. So essentially, this will potentially capture some dependencies of the gene. The genes that you can predict with high confidence will be added to your known gene list, and then you gradually grow the list in a way that you're training the model to learn the potential dependencies between these different genes. So I skipped this fine-tune part, and I will skip this SC foundation model, because I want to get to the last open question, maybe a couple more minutes on this. So first of all, I think it's still an open question how to better evaluate these large language models, different language models, right? They have different number of parameters, so it is difficult to really very fairly compare them. Oh, you can compare it to some conventional methods, single cell, all of them are comparing to SCVI or some other methods. But is it a fair comparison? Because large language model, you have pre-trained on lots of data. You have seen so many different conditions. Is it, you expect to do a bit better, but you expect to do how much better? I think that's the question, right? And right now, if the results always is comparable, a little bit better, whether it shows that the model is not necessarily learning the new things as compared to these conventional models. And certainly how to make these large language models accessible, right? Everyone wants to use this, but right now only a small number of groups can actually do that. And this is really I talk to my students all the time. How to embed cell gene to better maintain biological context, like whether the current tokenization or these representation is the right way of doing this kind of model building in the first place. Does a cell work that way? You have a list of genes and you order them that way, and or the DNA sequence you consider this k-mers, or is there better ways of doing this? how to incorporate some prior knowledge, right? You all know, there's dependencies, there's other very complex biological insights that we have learned from decades of research, how to incorporate them into these models. So I'll just list them all. I think there's one more. So yes, so for example, this one, right? So what are the important problems for large language? Are they solving anything that other models cannot solve, the conventional models cannot solve? I think at the moment, I think the answer is we don't know, because we haven't seen that yet. Can we actually combine information from literature, right? You can build a LLM for PubMed together with this LLM for genomic data. Maybe they'll have some amazing findings. And in fact-based field, people don't like hallucination, but maybe in biology that's a good thing to have, right? Because these reliable hallucination could generate very interesting biological hypotheses for us to evaluate further. So to summarize, this year actually marks the 70th anniversary of the publication of the DNA double helix, right? And it was published in 1953. And over 20 years ago, the human genome, draft genome, was published. And I was in China as a college student, and I was really excited about the DNA sequence and how to apply computer science techniques to that. And these days, I think there's also a lot of excitement in that. I personally think this might be another those moments that there might be some very big breakthroughs. But I think I want to say to the students, I think this is really an exciting time, and you're working on in an exciting field. And with these new developments, I hope more of you will be interested in exploring these methods and to find out the actual problems, important problems that these new development, new tools can solve. And you're really the ones driving this, right? As I have told you, your advisors not necessarily know more than you do for a lot of these things. So you're really the ones that are driving this. And then I think there's a difference between, you actually have written code to work on this, right? And just like me, we're just looking at reading papers. I always told my students the last time I seriously wrote some code, it was during the first term of Obama administration. So that was a long time ago. Yeah.
Do you think you should start writing code again?
Yeah, probably. There's a lot of these excitement in the field. This was a recent news article on Forbes. This is on protein structures, but I think some of what they talk about applies to other areas in genomics as well. Before I stop, I just want to advertise this because Recom 2024 will be in Boston, and I'll be serving the PC program chair. And then I just want to ask all of you to consider submitting to Recom. The deadline is October 20th, but you need to submit an abstract of your paper by 2016. I think I will stop here. I think I'm already over time. Yeah.
We may, it depends on availability, we may have a travel fellowship for a paper that starts here to go to Recom.
You said 2016. Is it really? Is it the date? I think it got us a bit confused.
What?
You mean October 16th?
I mean October 16. October 16. Yeah.
People got confused.
Oh, I said what?
You said 2016.
I said 26? Oh, no. October 16. Sorry.